right, so we will go ahead and get started. Are there any other kind of questions before we jump in today? All right, fantastic. All right, so this morning we are going to be transitioning a little bit from innate immunity to adaptive immunity. But before we do that, we have our visiting virus presentation this morning, and today it will be given by Joey. And you should be, all set to share your screen.
All right. All right, so for my visiting virus presentation, I did the Marburg virus. So the virus family is it's under the Filoviridae, which is actually the same family as Ebola. And as you can see on the right, they're actually like very similar in strands. Its nucleic acid content is a single-stranded negative sense RNA. It's very dangerous. It's got a 88% fatality ratio. And the reason behind it is because it causes hemorrhagic fever, which can interfere with blood clotting, which leads to severe bleeding. It causes multi-system organ failure, and it has the inability for the body to regulate itself through its prevention of blood clotting. So, like I said, it leads to severe bleeding. It originated in Africa, but it was first documented in Serbia and Marburg, Germany, hence the name Marburg virus. It was usually transmitted from fruit bats or megabats. They go by two different names. And it's from prolonged exposure in mines or caves. So basically they have to bite you or come in direct contact with you. The host tropism is in humans. And like I said, it's through direct contact through mucous membranes or, broken skin, whether it be a cut or a scrape or what have you. And it remains infectious until the virus is physically removed from the bloodstream. So here's here's a diagram that shows the pathology of the virus. So at first it's direct contact through exposure from the virus, and it can affect multiple organ systems, like I said. So on the bottom, you got your liver, and your liver, your spleen, your kidneys, and your blood vessels. And the big reason why it causes such huge damage across the entire body is from cell death occurring across numerous tissues, like your hepatocytes, your lymphocytes, your epithelial cells or tissue in your kidneys, and then the inability for the blood to clot through a cut. And then, so the overall importance, it's like I said, it's very easily transmitted. It's actually very common for healthcare workers to contract the virus through through direct patient care. And then it has a high mortality rate. So usually death can occur 8 to 9 days after symptoms begin. So it acts very quickly on the body. And it's also very identical to Ebola, and we all know how deadly that was when there was an outbreak. Also, there's very little treatment for it. Just like there's no cure. So the only really treatment they have available is rehydration orally or through IVs. And they're developing potential immune and drug therapies, but they're not at the level where the CDC believes it's like the best method of treating the virus. Also, it's very hard to distinguish and diagnose between other infectious diseases like Ebola because of how close the strands look. But there's also malaria, typhoid fever, and meningitis. And that's it.
Oh, fantastic. Did you mention why you ended up picking this virus?
Basically, it was a mix of, I kind of searched the most deadly viruses known on the planet. And also I never really knew about the Marburg virus. But what I found, it was very similar to Ebola, and I know how bad that outbreak was a couple of years back. I figured that it was good to touch on since they're so identical.
Yeah, they are. They often get not grouped together, but when we're talking about some of the most deadly viruses with that higher fatality rate, they definitely are grouped together, Ebola and this Marburg virus. Fantastic. Thank you so much, Joey. All right, so before we jump into our material for today, I do have a quick announcement, kind of a reminder. So for Monday, March 8th, your additional resource paper is due for your topic paper and project, all right? So all I'm looking for that is a primary research article. The research article, because it's a primary research article, should have a results section, it should have a methods section, and it should be some type of new research that's being published, all right? I'm not looking for another submission of a review paper Although if you find a review paper that you're really interested in, you're welcome to use that as an additional resource for this assignment. But what I'm looking for is the integration of at least one primary research article. All right. And so you'll just need to submit that article to Brightspace for Monday. I'll take a look at it, see if it looks like a good reference, see if it's a primary research article, and just kind of okay that. All right. So that needs to be submitted for Brightspace on Monday. And then next Friday, so a week from today, you'll be taking about half the class to meet with the other people in your topic group. Now remember, the written portion, that writing assignment, is done on your own, but the presentation at the end of the semester is done in groups. So you can kind of talk with your group about maybe what you find interesting about your topic so far, about any of the review papers that were posted on Brightspace, or kind of you can discuss the way you want to kind of tailor your topic paper. All right. So just kind of getting familiar with that topic in general and seeing your group members' thoughts on it. All right. And then for Monday, the 15th, March 15th, your outline of your writing assignment will be due. And we'll be going through more of that, or more about that outline, next week in class. So I'll give you some requirements for that outline. But I would like you to do an outline, because it's always nice to get some of those thoughts in order before you try and write a paper like this, where we have a lot of different topics and subtopics going on. Any questions on that?
When you said fairly new, do you have like a time frame of like years that you want the research article to be from? Or
Usually within the last 4 or 5 years. Yeah, so usually within the last 4 or 5 years, you want to shoot for it. And just because there's always evolving research, so while a paper from 10 years ago can still be valuable, it's nice to see where that research has gone from 10 years ago till now. All right. All right, so as I mentioned, we're going to be switching a little bit from our focus on innate immunity into more adaptive immunity, right? So mainly in this course so far, we've been talking about elements of the innate immunity or innate immune response. And you can see those there, right? Our phagocytes, our macrophages, our neutrophils, dendritic cells, right, as this messenger between innate and adaptive. We've also talked about complement, natural killer cells. These are all really falling into that innate immunity category, right? And so today we're going to be transitioning to start to discuss the adaptive immune system. All right. And specifically, we're going to start with B cells and antibodies. So in our introduction so far to our adaptive immune system, right, we know it's made up of B cells and T cells, or B lymphocytes and T lymphocytes. And these are part of that adaptive immune response. They're going to provide that more specific response to pathogen. And when we talk about our B lymphocytes, they start off as a B cell. They then mature to what is known as a plasma cell. And then we're going to have the production of antibodies from those plasma cells. And antibodies, as we know, we've briefly mentioned them in this class before, right? They have the ability to label that pathogen and help the immune system clear that pathogen from the system. And so today we'll be talking about the basic shape of our antibodies and how they bind those pathogens in general. All right. So what do we know, and maybe what have you heard about our antibodies? We just kind of gave that broad introduction, right? They're able to identify pathogens, but what else do you remember that we've maybe talked about, or that maybe you've heard now in the news about why our antibodies are important?
They contribute to that like immune memory that helps us if we have like a secondary infection to help target that infection so it doesn't like cause any sort of bodily harm.
Yeah, right. Another aspect of why antibodies are really important is this ability to stick around in our system and last and provide that immune memory. So if we get infected with that same pathogen, We don't have to activate that immune system, wait for that immune response. We already have those antibodies present and can make those in the system. So we can clear that pathogen much easier than we would after that first infection. Anything else? What about what a typical antibody kind of looks like? Do we know any of the characteristics about that?
It's shaped like a Y, and the like Y portion is like out like towards like the outside of like the membrane, and like the little like stalk is attached to the surface membrane.
Yeah, exactly, right? They have that classic kind of Y shape. And the parts of that Y, as you've kind of mentioned, right, they're going to play different roles in the functionality of that antibody. Fantastic. So in our overview of antibodies, we have to start with where they come from, right? And so in this image, we're really looking at this progression of how B cells start to develop and how they then mature to eventually producing antibodies. So we start with what is known as a resting B cell. This B cell has developed, it's been released into the body, it's now ready to look for pathogen, but is still known as this kind of resting B cell. What you'll notice about this resting B cell is it has membrane bound what are known as immunoglobulins. And so the general term for B cell receptor that we see on the surface of our B cell is immunoglobulin. It's able to bind pathogen. And then once we see that B cell binding pathogen, once that B cell encounters pathogen, right? Or more specifically, right, an antigen that's found on that pathogen, right? We see this looks like some sort of cell surface protein on the bacteria. These antibodies are able to recognize that cell surface protein. And this is what's going to stimulate this B cell to then become a plasma cell. And it's when we have that plasma cell present, right, when it's already been activated by pathogen, that now that cell can start producing secreted antibodies that can go out into the system and help fight against that pathogen. All right. And this process of a B cell identifying an antigen, of being selected for because it can identify that antigen, is known as clonal selection. So we mentioned way back at the beginning when we did this overview of the parts of the immune system, and we briefly talked about adaptive immune system. We mentioned how that adaptive immune system, those lymphocytes, have to be activated by pathogen, right? And because they're so specific, this activation is really important. We only want to be activating B cells that are going to help us fight that pathogen that we are dealing with at the time. And so this idea of clonal selection is this selection process that stimulates B cells that are gonna identify that bacterial pathogen to be selected for and to start secreting those antibodies. So it is this selection process. Only B cells that are producing antibodies that can identify the pathogen the host is trying to fight will move on to becoming plasma cells. Right. And that's because those are the cells that are going to produce antibodies that can recognize the specific pathogen that's being dealt with at the time. All right. So I just want to share a few kind of common terms that we'll be using as we move throughout this section of immunology. So as I mentioned, immunoglobulin is the general term for either the B cell receptor, so this membrane bound form of our B cell receptor, or the secreted antibody. So we could call these little Ys over here that are attached to our B cell immunoglobulins, and we could also call these secreted antibodies immunoglobulins. It's more of a general term. And then antibodies, will refer to those as the secreted form over here, right? So once those B cell receptors, once those immunoglobulins are released from that cell membrane, when they are secreted, this is when we start to have when we can start to use the term of antibodies. Right. And so as we've mentioned before, right, antibodies are considered really specific. Our adaptive immune system is much more specific than our innate immune system, right? Our innate immune system is just trying to figure out what type of pathogen is present, right? Is it viral, is it bacterial, and trying to clear that type of pathogen. But when we have activation of our adaptive immune system, this is much more specific. And one of these reasons is that specificity of these antibodies, right? And so as we can see in this image, we see this B cell receptor, this immunoglobulin, binding to a certain cellular surface protein on that bacteria. This antibody is only going to be able to bind that specific antigen, that specific cell surface protein. It's not going to be able to bind the whole bacteria or the whole virus. It's going to be very specific for a certain element or antigen of that pathogen. Right. And so this usually is going to include, like I mentioned, these cell surface proteins. It can also include carbohydrates. And everybody, because these antibodies are so specific, I think this is really pretty cool to think about, right? Everybody has their own repertoire of antibodies, their own collection of antibodies that because of the pathogens they've come in contact with, they have their very own repertoire or collection of those antibodies, right? So it depends on what pathogens you've been in contact with before, and that's going to affect what antibodies you carry around with you.
I have a question.
Yeah.
This might just be like because like mutations and stuff, but if like with what you explained, like how come people can get sick with the same thing, like strep throat or something like that, like again and again, like constantly?
So that's going to come down to maybe slight differences. Strep throat is caused by a bacterial pathogen. So there can be slight differences in how that pathogen is recognized. We're actually going to talk a little bit about that later on today when we bring up how that antibody actually binds, and we'll be talking about it with the example of these different COVID-19 variants, right? What that really means and how those are able to avoid the antibodies that we've produced. So we will get a little bit more into that, but it does have to do with this binding of the antibody, and if the pathogen can somehow interfere with that or change that area where the antibody binds. Additionally, just kind of as I want to mention here, although we'll talk about vaccines in much more detail later on in the semester, the most successful vaccines are the vaccines that can produce antibody production or produce antibodies. So they initiate or activate those B cells, and those B cells then become plasma cells that can generate those antibodies against whatever pathogen we're trying to vaccinate against. So now we'll really get into a little bit more about antibody structure. And there are a number of ways that we can label the different pieces of our antibody. All right. So in this first categorization, we can split up this antibody structure into what are known as light chains and heavy chains. All right. And so in this image, what you're looking at is the light chain shown in yellow. And then in green, we have the heavy chain. All right. And you can see that classic Y shape, right? We have the top of our Y here, and then kind of the bottom or stalk of the Y down here. All right. We also have the addition of certain carbohydrates that can attach to this protein. And so the way that we get these names, heavy and light chain, is due to the size of these protein chains, right? So when we're looking at antibody structure and when we're looking at a diagram like this, I want us to remember that this is a protein structure, right? And so this Y is made out of light chains, which is that certain subunit or certain protein. And then we have heavy chains. And that light chain weighs less than the heavy chain does. So that's where those names came from. You can see here that in this diagram, right, we have two kind of pieces of our light chain, And one heavy chain is going to be a piece here, 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 and here. All right. And so a heavy and light chain, you can see this little connection here. We have a disulfide bond that links these two chains together. And then a heavy and light chain comes together with another heavy and light chain. And that's where we get this classic Y shape. Right. And you can see here the two, if we take this as one heavy and light chain, right, we have this connection between our other heavy and light chain through these disulfide bonds. All right, and those are typical bonds that we'd see in protein structure. We can also delineate the antibody structure between what are known as the variable and constant regions. So when we're talking about the heavy and light chain with this yellow and green, we're really speaking more toward the overall protein structure. When we start talking about the regions, the variable or constant region, we're really more speaking to the function of that antibody. So here we can see our constant region shown in blue down at the bottom of our Y. And this constant region is really, really important to interact with cells of the immune system. So this is the part of our antibody that'll work with our macrophages or other cells of the immune system to help clear that pathogen away. Up at the top, we have the variable region shown in red. And as that might suggest, this is the variable part of the antibody. And it's the part that recognizes pathogen, right? And because we have so many pathogens that potentially our immune system has to recognize, if this is going to be that variable or different region that we see between different antibodies. All right, so our functionality of our antibody really comes down to these different regions. The constant region is going to interact with cells of the immune system, and that variable region is going to recognize the pathogen. Now, what I also want to point out is, before we get to this hinge region, I also just want to point out, now that those boxes are gone, that the variable region is where we have our antigen binding sites, right? So that's the part of the antibody that's going to bind to the pathogen itself. What we also have to point out, though, is this hinge region. And this hinge region is what connects these two heavy chain and light chains together, right? It connects our antibody to make that full Y, right? And so this hinge region is really important because it allows for the flexibility of this antibody. And so if you look at this little bacterium over here, we have different antigens on that cell surface, right? Some of them have this circle, some have a rectangle, triangle. These are all representing different potential antigens that an antibody could identify and bind to, right? And so here, the hinge region is really important to allow flexibility of that antibody and to allow for that antibody to bind an antigen multiple times on the cell surface. Right? So another thing I want to point out is that we're going to have two antigen binding sites, right? We have two variable regions. We're going to have two antigen binding sites. And so you can see in each of these cases, this antibody is able to bind the antigen it recognizes twice on that bacterial surface. What do you think the advantage is for an antibody that can bind not just one site, but two sites?
It has a broader range of function.
Yeah, potentially, right? Maybe it could have a broader range of function. Typically, we're going to have an antibody recognize two of those same sites, right? So what allows for not just the functionality of the antibody, but how is one binding site not as effective as two binding sites?
Would two binding sites make the, the overall like bond strength stronger?
Yeah, right, exactly. If we have an antibody that's bound this pathogen twice, it has a much stronger bond to that bacteria, to that pathogen, right? And so if we can bind this bacteria twice, it really makes sure that that antibody is bound tightly and that it's not going to be released or have that bond be broken. Right? And so the way that this antibody is set up with this hinge region allows for a higher chance of this antibody binding twice to a bacteria, right? So maybe those antigens are really close together, or maybe they're a little bit more spread out, like this bottom example. And that hinge is going to make it possible to increase that chance of tight binding with both antigen binding sites. All right, any questions before we move on? All right. So the antibody that we've just been looking at, that we've been learning about the structure of, is known as an IgG antibody. But we have different classes of antibodies in our bodies that play different roles in the adaptive immune response. All right. So here we're again looking at these antibodies have been colored by that region, right? So you can see that variable region up here, and then we have our constant region down here. But remember, we also have these light chains on the outside, then kind of the very outside of our Y, and then we have these heavy chains on the inside. And the different classes of antibodies are going to differ in their heavy chain. So this inside chain of our Y, we have different classes of antibodies that have different heavy chains. And these differences in heavy chains differ between the class, or what is known as the isotype of the antibody. So the one that you've probably heard of, or the one that you've seen most, is going to be this IgG antibody. But we can also have what are known as IgM antibodies. And as you can see, these have an additional kind of segment here, right? We have IgD antibodies, which you can see have many more carbohydrates than compared to our IgG. IgA, again, we see differences in carbohydrates, but also if you look closely, that hinge region as well. And then lastly, we have IgE. And because these isotypes differ in heavy chain, right, in the chain that is found towards the bottom of our antibody, right, these isotypes are going to interact with our immune system in different ways. And as we move through this discussion of adaptive immunity, we'll discuss how these different types of antibodies are found in different locations in our body and how they play different roles in clearing pathogen in different ways. All right. So as we talk more about structure and as we continue, we'll mainly be focusing on IgG, but I wanted to introduce you to this idea of our different isotypes of antibodies and the fact that they're going to be different in their heavy chains, all right, and in the structure of that heavy chain. So as a reminder, and just to give you a little bit more of a visual here on your antibody structure, remember this is a protein, right? So we're looking at diagrams of these regions. We know it has this general Y structure, but we can also take a look at that protein structure. So what you're looking at here on the left are the different pieces of that antibody, or the different protein structures of the antibody. So if we look over on our diagram here, right, we see that in purple, or in this kind of purplish blue color, right, we're going to have our heavy chain, right? Our heavy chain makes up that inside portion of that Y, right? And then part of the top part of that Y. And so you can see here these different kind of domains of our protein structure that form that overall Y. In yellow, we have the other heavy chain. And then in pink here, we have that light chain, right? And so you can really see how that protein structure mimics this Y shape, right? What I also want you to notice, which is highlighted in this middle image, but we'll also discuss down here, are the ways that these different pieces of antibody structure or protein structure are delineated or labeled. All right. So when we talk down here about our constant region, there's a constant region, right, shown in blue, and this is going to be part of our heavy chain. So very often it'll be delineated C for constant, and then heavy 1, heavy 2, or heavy 3, right? So that's indicating that these pieces of our antibody are from that constant region and they're part of that heavy chain, right? And you can see them labeled up here as well in this diagram. When we move over to the light chains, right, we do have a part of our light chain that is constant. So this would be known as CL, right? Constant light chain. And then we can get down to the variable part of that antibody. And both the heavy chain and light chain have variable pieces to it, right? Have regions that fall into this variable region. And so here we have the VL segment of our antibody and the VH, right? Variable light, variable heavy. Any questions on that? All right. And then lastly, it's always important to remember that where we have that variable region, this is where we have those antigen binding sites, right? Where that pathogen is going to come in contact with that antibody. All right, so this leads me to a couple of review questions that I want to pose here. The first is about the 5 different isotypes of immunoglobulins. So how do these differ from one another? I'll launch a poll here. All right, take about 10 or 15 more seconds, try and get your answer in. All right. So we had some answers for B, C, D, and E. The majority of you, however, said B, and this is the correct answer. So our correct answer here would be B, the heavy chain constant regions. So as we could see when we were walking through that diagram of the different isotypes, all of them needed that variable region, right? That's the region that's going to interact with the pathogen, right? All antibodies need to be able to interact with that pathogen and recognize that pathogen. But where we get the difference for our isotypes is in that heavy chain constant region. So in that kind of base or bottom part of our antibody. And that's going to mean differences for how those immunoglobulins interact with the immune system. So isotypes really focus on, right, those different classes of antibodies really focus on how the immunoglobulin is going to interact with the immune system, right? How that antibody is going to interact with those cells of our immune system. And so now I want to focus a little bit on how our immunoglobulins kind of are diverse, right? How they recognize all of these different types of pathogens. And so in this case, we specifically need to talk about our antigen binding site, right? So this antigen binding site that's found in these variable regions that is going to allow for the antibody or the immunoglobulin to bind pathogen, all right? And so over here, we're looking at a number of different ways that we can look at the light chain variable region. So in this case, we're taking a look at our light chain variable region, right? That would be here in our antibody or over here, right? And in our very bottom image, you can see that the variable region of this light chain has been split up to kind of represent the majority of that region, and then also to highlight the specific piece of that will be that antigen binding site. And it's here in this antigen binding site that we see very, very high variability in the identity of the amino acids that are found in that antigen binding site. So if you look up here on our top image, you can see this is representing that protein sequence. And these yellow and red are corresponding. The red you can see is where that antigen binding spot is. And on our y-axis over here, we're looking at the variability in the amino acids that are found in this part of our protein. And so as we see these big spikes in variability, these are corresponding to where we see antigen binding to the antibody. And this is all to really say that there are areas of the antibody that are highly variable, that differ in protein structure and amino acid identity, because different amino acids are going to create a different antigen binding site that's going to be able to bind to a variety of different pathogens, right? We need high variability in that antigen binding site to accommodate all the different possible pathogens that this antibody or that a person might run into, right? And so in this middle section, you can see that protein structure again. So here we're looking at the protein structure of that variable region on our light chain. And you can see these pieces at the very end are where we see those red pieces, right? Those areas of the protein with really high variability, with the ability to change to identify those different pathogens that we might encounter. And so these are known as hypervariable regions, right? Very variable regions of the antibody, or they can also be referred to as complementary determining regions, right? The regions that are going to determine what type of antigen this antibody can bind. Any questions here? All right. So this leads me to another couple review questions, just so we're all on the same page with this terminology. All right, so I'm going to launch another poll. Which of the following corresponds to the antigen binding site of immunoglobulins? All right, take another 10 or 15 seconds, try and get your answer in. All right. So I had answers for all options here. The majority of you said B, and B is our correct answer here. All right, so when we're looking at the antigen binding site, right, that antigen binding site is where we have, or is found in, the variable regions, right? And that's going to be the variable regions of both our heavy and our light chains. And so our only option here, right, V is standing for variable, while C is standing for constant. And so our only option here that includes both of those variable domains is going to be B, the variable domain of the heavy chain and the variable domain of the light chain. All right, I have one more review question here. Again, speaking to some of this terminology that we've just worked through, another term commonly used to describe hypervariable loops is what? All right, 10 or 15 more seconds. All right. So we did have answers again for all of these, but our majority answer was D, and that is the correct answer. All right, so hypervariable loops or hypervariable regions is synonymous with complementary determining regions. All right, those regions at the very end that are making up our antigen binding domain. Many of you also said hinge region as your answer for this, and that, remember, is that way that our antibody is able to open or close the top of the Y. All right. So this hypervariable region or this complementary determining region is what is going to make up our antigen binding domain. All right. So now I want to talk a little bit more about this variable domain and about the way that antibodies can bind to different types of pathogens. Right? So these antigen binding sites that we just talked about in that complementary determining region or in that hypervariable loop are going to differ in the identity of their amino acids in that region. And that is going to have a result or an effect on the different shape and physical properties of that antigen binding site. All right. And so the part of the antigen that an antibody can bind to is known as the antigenic determinant or the epitope. So we're getting kind of more and more specific on how we refer to that part of the pathogen that an antibody can bind to, right? So a pathogen will have different antigens, and the piece of that antigen that an antibody can bind to is going to be known as the epitope, right? And so an antigen can have different epitopes. So a single antigen can have different areas that different antibodies could bind to, or an antigen might just have one type of epitope that is repeated. Right, so we can see that difference here. And so here, this red is representing just a piece of that pathogen, right? We're looking at the antigen here, and that antigen is going to have different binding spots that are either different, right? We see 4 different epitopes here, or the same and just repeated, where we see this one epitope repeated that allows for multiple places where that antibody can bind. And so here, these would both be referred to as multivalent antigens. That means that this antigen has more than one spot where an antibody can bind, and these can either be different, as we see in this example, or the same, right? So what I want you to really kind of pay attention to here is that we've talked about pathogens. We've talked about how those pathogens are going to have antigens, right? That foreign piece or that foreign trigger to the immune system. And now we're talking about that specific piece of antigen that an antibody can come in and bind to. And that's going to be known as an epitope. So when we talk about this in terms of our SARS-CoV-2 virus, right, we have the pathogen itself, that viral structure itself, and we've talked about some of the surface proteins that this pathogen has. Specifically, the one that the vaccine is trying to elicit an immune response against is going to be that antigen of this SARS-CoV-2 pathogen. And that antigen that they're focused in on is that spike protein that's being shown in red here, right? So we have our pathogen, SARS-CoV-2. We then have the antigen, which is this spike protein. And now we can break it down even more to the epitope of this. And that's where that antibody is specifically going to bind to, the piece of that spike protein that that antigen binding site is able to recognize, right? And so if we take an even closer look at the spike protein itself, you can see kind of a side view here, and then kind of a top-down view of that spike protein, right? And so the different areas of this spike protein could have different epitopes, different places where an antibody can bind, right? So we take a look at this, we could have an antibody that binds over here, over here, over here. Those are all going to be different epitopes that are found on the antigen. Any questions here? All right. And so now is where we start to see if a virus, right, if this virus is able to change these specific epitopes, these areas where those antibodies can bind, this is where we run into issues with immune memory or having that repertoire of antibodies recognize that pathogen again, right? If you get sick with a certain virus, your antibody is going to recognize a specific part of that virus. And if the virus can mutate and evolve to then change that area, we're going to need a new antibody to recognize that pathogen.
I have one quick question actually.
Yeah.
On that last slide, so like can 3 antibodies like all like simultaneously bind to this protein on like the 3 various epitopes, or just like does one antibody bind to one site and then it basically like, I guess like claims it as like being like attached, if you know what I mean?
Yeah, I do. So it kind of comes down back if we look at kind of this image, it depends on that antigen. And it also depends on how your immune response is triggered, right? We'll learn more about how these antibodies are formed, but we first need a B cell to recognize one of these epitopes, right? It has to be activated by the pathogen itself. And so we could have some B cells that are being activated, they're going to produce all the same antibodies, right? And so we'll have that one kind of type of antibody in our system. Now, if our immune system can be triggered so that we're activating multiple B cells against the same antigen, we can have variation in that antibody that's able to bind. But it depends on that activation of the immune system and how those antibodies are produced. But just like we see in this image, we can have multiple antibodies being able to bind to an antigen. It just is kind of pathogen specific. Yeah, great question though. All right. I'm going to just skip this a little bit. I know, are we just about out of time? We're out of time. All right. So I have posted a couple of videos about antibodies on Brightspace. I was hoping to show one today, but we just don't have the time. So take a look at that. It does go through this protein structure in a little bit more detail and in a more visual way. So you can absolutely take a look at that. On Monday, we are going to continue with our discussion on how the variability of these antibodies arises. All right. And for that, you're going to need to understand a little bit of molecular biology. So if you're rusty in how DNA becomes protein, I've posted a bit of a review on Brightspace as well. So you can take a look at that. We will be going over it briefly in class, but if you know that you haven't thought about how DNA goes to RNA, goes to protein in quite some time, I would take a look at that as well this weekend. All right. But you have no review questions, so you don't have to worry about that. And I will see you all on Monday. Have a great weekend.
Thank you.
Thank you.